Hi everyone, welcome to my channel Pure Biology, and today I come with another video, that is the bacterial recombination or the transformation of the DNA, how it's occurring in bacteria. And there are three processes we have to learn about this bacterial recombination or gene transformation, that are the transformation, conjugation and transduction. So first topic, that is the transformation. Okay, and before going to the topic, I want to say that sorry for not giving any video for a long time, because I was busy for another work, and that's why it was impossible to me for doing this. Okay, so again I want to say thank you to all of you who have already subscribed my channel and support me a lot. So let's start. That is the transformation. So the transformation is what? This is the horizontal gene transfer process. This is the horizontal gene transfer process. Okay? And sorry. So this is the horizontal gene transfer mechanism, and this is first discovered in bacteria, and who discovered? The name is Griffith. Okay, Fred Griffith. Fred Griffith. In 1928. 1928. Okay, Fred Griffith in 1928. This is the uptake of a DNA molecules or the fragment from the medium by a cell, and the incorporation of this molecule, or you can say the fragment of DNA, into the chromosome of the recipient. So here the two bacteria present. One bacterium is called the donor and another bacterium is called the recipient. You all know that. And here the transformation when occurring, the donor generally transfer the gene fragment into the recipient body or recipient cytoplasm. Generally, this may be natural and artificial. So natural, this is very rare and found in Streptococcus pneumoniae and Haemophilus influenzae. The Streptococcus pneumoniae, these are the gram-positive bacteria, and Haemophilus pneumoniae, that is the gram-negative bacteria. And these bacteria are very important, or these are involved in this transformation process. The ability of a recipient to take up free DNA and become transformed is known as competence. Okay, and this natural and artificial process, natural means you know that is occurring in the environment, but artificial, that is occurring in the lab or laboratory or some experimental process. We can do this transformation process. By transformation process, we try to inject DNA from one bacterium to another bacterium. Okay, so here what are called the competence? The competence are actually the recipient bacteria who have the capability to take up the free DNA and become transformed. These are called the competent bacteria, and it is an inheritable characteristic, or you can say that inheritable feature of some bacteria. That means one bacteria, if one is competent bacteria, that means its next generation may be competent bacteria or sometimes not also. And this competent bacteria that can take up DNA, encode proteins. Okay, they have one kind of proteins. These competent bacteria, they have one kind of proteins. These proteins are called the competence factors. Competence factors. And what are the function of these competence factors? These proteins generally facilitate the binding of DNA fragments to the cell surface and uptake of DNA into the cytoplasm. That means, what is the actual function of this competence factor? They facilitate the binding of DNA fragments to cell surface. Actually, by binding the DNA, free DNA, that on the to the cell surface and intake within the cytoplasm. Okay. Now, one strain, that is the one second, Haemophilus. Okay, this is the Haemophilus, HAE, Haemophilus species. So here in Haemophilus species, the DNA uptake is associated with the formation of small membranous structures, or these are called the transformosomes. Okay, transformosomes. So these transformosomes, which protrude outside the cell, and the transforming DNA is taken into these vesicles, where it is when internalized into the cell. So what is actual work or function of these transformosomes? These are the small vesicles or the sac-like structures. Okay, these are very small vesicles or the sac-like structures, and their function is that they help in the internalization of the gene or the DNA, or you can say that DNA internalized within the cytoplasm through these vesicles. Clear? Sometimes this bacterial strain, another type of bacterial strain of this species, obviously they have some sequences, and these are the very specific sequence, or you can say that uptake sequence, and these uptake sequence are very important for internalizing the gene or the DNA fragment. Without this uptake sequence, they cannot internalize the DNA fragments. So maybe they have small vesicle-like structure, or the specific sequences that are involved for getting the DNA fragments within their cell, or internalizing the DNA fragments within their cytoplasm. Okay. Next, the process. Here I just try to explain about the natural process, because the artificial process is totally done by the man in laboratory. So here we will just learn about the natural process. So what actually happened in the natural process? Here the competent bacteria actively pull the DNA fragments into their cells, and DNA can be naturally released into the environment when bacterial cells die and subsequent lysis. In some, that is actually saying that in some lysis process, when the bacteria that become die, and in that situation their DNA remain within the environment. And so in that situation, what is the function of this competent bacteria, or what can they do? This competent bacteria, they just take these released DNA fragments from the died bacteria. Okay, you can say that what is happening here? Again I repeat that when the lysis process is occurring, so bacteria may they died, and what happening? The DNA fragments that remain in the environment, and in that situation the competent bacteria, they intake that DNA fragments that remain within the environment. Clear? So once free DNA comes in contact, and it is happening only at that situation when the free DNA that present in the environment when come in contact with the competent bacteria, or you can say that competent bacteria reversely that may be contact with the releasing DNA or the DNA that remain in the environment. Clear? So linear dsDNA that enters within the competent bacteria, and one strand, which strand entering within the bacterial cell? The strand that is the leading. You all know that DNA have the two strands, leading and the logging. Sorry. Okay, so here the leading strand that is translocated, but the lagging strand that not translocated and is degraded by the nuclease enzyme at membrane surface. So we all know that DNA has the two strands. One is the leading strand and another is the lagging strand. So only the leading strand that enter within the cytoplasm of the bacteria, but another strand, that is the lagging strand, that is degraded by the nucleus at the membrane surface. And the translocated DNA that integrates with the chromosome by the homologous recombination, and the recipient cells undergo this process and acquire a new phenotype. So what is happening here? The translocation is occurring by the process of recombination, and here the two which type of recombination? This is a homologous recombination, and here the new character is, or new phenotype is seen within the not new phenotype is seen, you can say the new character is seen. Okay, so recipient cells with the new character that acquire a new character, and it is called now a new phenotype, or you can say that we will see the new phenotypic bacteria with the new character that is present or that is formed within the environment. So this is the transformation process. The natural transformation process I have explained here, and another important thing I have to explain you just after rubbing this one. So you just continue with this chapter. Okay. So here the single-stranded exogenotes are unstable and will usually be degraded unless they are integrated into the endogenote. So what are the exogenotes? Exogenotes are the donor bacteria. They try to donate their DNA into the recipient, and endogenote means they are the bacteria with the donor DNA. Okay. So if single strand is there within the exogenote, so they cannot unstable. They are very unstable. They cannot stable in the environment, and say that's why they try to donate their DNA within the endogenote. And by the process of what? By the process of homologous recombination. And if exogenote that contains an allele of the endogenote, the resulting recombinant double helix would contain one or more mismatched base pairs. So what is happening here? If exogenote that contains an allele of endogenote, if exogenote they have one allele just like endogenote, in that situation, after the recombination, the double helix or the recombinant double helix, they have one or more mismatched base pairs. Clear? One or more mismatched base pairs. In this situation, these are called the heteroduplex. These are called the heteroduplex. And the mismatches, these are repaired by the DNA repair enzymes. So the enzyme systems that replace the base in the new strand and thus restoring the genotype of the recipient. So its function. What is the function of the enzyme system? They just repair the base pair by replacing them, and in that way they restore the genotype of the recipient. And if the region is replicated before it is repaired, one of the two daughter chromosomes that will be recombinant and the other will be identical to the recipient. So without repairing the DNA, the replication is occurring. In that situation, one of the daughter chromosome that be recombinant and another that is the identical to the recipient. And here this is the very brief rough structure of the transformation. Here you can see that this is the transforming DNA and this is the bacterial chromosome. Okay, this is the bacterial chromosome, or DNA, and this is the transforming DNA. Okay, and this is the binding protein. Okay, DNA binding protein. If there is any noise, you can listen in this video, so please ignore that. Protein. Okay, so this is the protein where the DNA, the transforming DNA binds with this protein for the entering into the bacterial cell, and you can see that here the DNA try to bind with this protein, and after that the transforming DNA that enter within the bacterial cytoplasm, and the here the recombinations just before the starting of the recombinations, the position of the transforming DNA and the bacterial DNA or the recipient DNA. Clear? So this is the process of the transformation. In the next video, I will try to explain about the conjugation or the transduction. Okay, so you just continue with my channel and please support me for the growth of this channel. Thank you.